In this video, I'll be going over how to identify shrews, order Eulycotyphla, family Cericidae. So all of the shrews in Montana are in this family, within the order Eulycotyphla. This is the red-toothed shrews, which when you look at the skulls will become obvious why we call them that. The teeth are pigmented red along the tip, not the part by the gum line. And that's just one of the few things that are really distinctive about shrew skulls. Another is the unicuspids. So rather than the typical incisor, canine, premolar, molar, we see one single upper incisor that has two prongs to it. And then we see a series of about 5 unicuspids. They're called unicuspids because they just have one single little point. They're very simple little teeth. Now I say we have 5, but some of them are too small to be seen. So depending on the species, you will either see 3 or 4, and then the other ones are just so tiny that we don't really need to bother about it in this class because they aren't important for identification. And another thing about shrew skulls in general is you'll notice that they don't have a zygomatic arch. So that bone that kind of connects around in all the mammals we've seen so far is not completely formed. So to look at these skulls, we're going to look under microscopes or have really nice high quality photos taken because these skulls are all very small and the teeth are hard to see, which is why I'm standing here and I'm just going to go through my very crude drawings. So if we're seeing a shrew skull, and we've figured out that it is a shrew based on the unicuspids, the weird incisor, the lack of a zygomatic arch, and the small size. Now, which shrew is it? So there are 2 ones that you can knock out pretty easily. First one, Blorina brevicata. This is our short-tailed shrew. Has a very strange first incisor. I don't know if my drawing does it justice, but if you look on page 23 of your book, you can see it has a really massive first incisor with a big first prong and a very small second prong in comparison to, say, it's hard to see in black and white, but the other ones have a much more prominent second prong and less of a hefty curved first prong on the incisor. So that makes it pretty clear that it's Blorina brevicata. It's also quite large for a shrew. So CBL over 18.5. This is, I'm gonna just grab this right here. Because this is one that is possibly viewable. So this is maybe a third longer than all the other shrew skulls. This is, by shrew standards, massive. So that's Blorina brevicata. The other one that's pretty easy to identify is Sorex hoyi, which only has 3 visible unicuspids. So you see the incisor. Don't be fooled by the second prong. That's not a unicuspid, it's part of the incisor. And then we see 1, 2, 3. All these other ones have 4 that we can see. So if it has 3, Sorex hoyi, you figured it out, you're done. Now for these other ones that have 4 unicuspids visible, we're going to look at the 3rd and 4th unicuspids. And in these 2, the 3rd unicuspid is about the same size, maybe a little bigger than the 4th. So we can see in my drawing here, and you can see in the key that has some photographs, and you can see on the photos online. Looking, this is a side view, so we see incisor and then these 4 unicuspids looking at the 3rd and 4th about the same size, or maybe 4th a little smaller. For these other ones, the 3rd will be smaller than the 4th. It might not be a lot smaller, so you have to pay attention, but if you look closely, you can see the 3rd one is smaller than the fourth one for these other ones. So third equal about to fourth. How do we tell these two apart? So then if we look at the shrew skull kind of head on, so right now we have been looking from the side. If we look instead straight on, like face view, we can see the two upper incisors. And what we're looking at is when we see these two upper incisors head on, is there a bump on each incisor that kind of joins together and makes contact? That bump is called a medial tine. Medial in the middle, tine, like the points on a fork are called tines. So this is a little bump on the incisor when you look straight on. Psoas mariami doesn't have that bump. If you look between the two upper incisors straight on, it's smooth. But Psoas cineris does have bumps. So we can tell these two apart. Now, looking at these last four, so we've already figured out that it is a shrew, we figured out that it has 4 unicuspids, and we figured out that the third is smaller than the fourth. Next step might be to see how big it is, because Psoas palustris, this water shrew, is quite large for shrews, CBL greater than 18.5. And if you wanted to look at the medial tine, you might not be able to tell that it's there. It's obscure. It is there, but depending on the coloration, the angle, etc., might not know. I would tend to go with the size. So then we have these 3 regular shrew size. We have tines. Now we're looking at the pigment line. So I mentioned that the teeth are red colored below a certain point. So for these 2 species, when you look head on those incisors, you can see that they're red colored up until about the point where the medial tine is. The pigment line might be below the tine, it might be just a little bit above the tine. My red here is kind of representing the rust red color you would see on the actual teeth. But on this monticolus, the tines are well below the pigment line. You don't see the pigment line kind of meeting the bump. It's way above. The bump is totally red. Almost there. Got these 2 that so far are identical. So then we'll use size. Psoas nanus is smaller than Psoas vagrans, the CBL less than 15 versus over 15.7. A note on looking at these skulls. So you'll want to get a side view. You may need a front view. If you're coming in person, the way we're doing this, is with these little tubes. So here's this tiny shrew skull. And you can look at this under the microscope by kind of tapping it into place and getting a good side view, or opening it up and looking at it straight on to get a front view of these incisors. If you're looking online, we should have nice photos of these, hopefully. And if you're in person, we have microscopes set up so that you can look at these more closely. So the last thing about shrews are the skins, which is relatively easy for us because we're going to avoid the really difficult stuff. So we have 2 species that you can identify the skins. One is Blorina brevicata. As I mentioned with the skull, it's really large as far as shrews go, and has a very short tail. Brevicata, brevi, short like brevity, cauda meaning tail, like a caudal fin. So I have the tail is less than a quarter of the body length, if you compare it to another shrew, you can see just how crazy short the tail is. Enough that you don't really even need to measure. And it is big. The other one that we can identify, Sorex palustris, is quite large compared to shrews. And it has very strong counter shading. So what I mean by that is the top of it is dark, the bottom of it is light. We can see this kind of darkish brown on top, and this dark grayish on top, whitish on the bottom. Now you might also, depending on how good of a view you get, be able to see these hairs coming off of the foot. It has these stiff, elongate hairs that it uses kind of light webbing, helping it paddle through the water. All the other shrews, well, we can't really figure them out. Here I have Sorex species. SPP here SPP period means any number of species. So I'm not talking about any Sorex in particular, I'm talking about the genus as a whole and various members of that genus. So all of these top few, not counting the bottom three, are species, mostly the species that we're covering. And you can see that they're all much smaller. They have relatively long tails. They're all brown or gray. They look like regular shrews. So we're going to leave it at that.